Morgen. Day.
So what I'm going to talk about today is experiments we've done to watch single molecules. So I thought I'd start just showing you an image. This is the bottom of a T cell, and these are T cell receptors, individual T cell receptors diffusing around on the surface of the cell. So the capability to make these type of measurements, imaging single molecules and live cells, only became available in the mid-90s, coinciding with when I came back and started my academic career at Cambridge. So basically what I've been doing over the last 25 years is developing these methods and trying to apply them insightfully to biology. And so I'm going to tell you a little bit about what we've been doing in that area. So what I'm going to do is basically introduce a little bit how we do these types of experiments, and then I'm going to talk about just two examples, how we've been developing and applying these methods to study protein aggregation and neurodegeneration, which I think is very interesting, just showing how these methods can start to tackle complex disease. And then the best example of application of these methods, which is actually one of the things I did very early on when I arrived in Cambridge, which was developing a so-called next generation DNA sequencing method. And really the main message, if you fall asleep in the lecture, is that new physical methods can enable new science. So just to make sure everyone's on the same scale, this is a cell, so it's tens of microns in size. This is a bacteria, this is a mitochondria. And there's a fundamental limit where diffraction limit, below which an object such as a virus or a protein is blurred by diffraction. So this protein, 10 nanometers in diameter, if we observed it by light, would appear to be 200 nanometers in diameter. And so I'm going to use the word nanometers. We're going to talk about molecules that are about 10 nanometers in size. We're going to be talking about cells which are tens of microns in size. And I'm going to be talking about methods, not that we developed, but we've been applying to break the diffraction limit, so we can actually see the structure of cells not at 200 nanometers resolution, but down at 20 nanometers resolution. So the first thing we normally do is build the instrument. So our labs are in the basement in the dark, with very colourful, as you'll see, with lots of different colour lasers. So this is Alex, building our instruments. So we build them on these very stable laser tables. So there's a whole bunch of optics which we use to manipulate the light. So we have different colour lasers. They all come over to a microscope. Hopefully you can see it here. And then we excite the sample, and then we have a very sensitive camera to image the fluorescence. So what I showed you at the start, and I'll show you again, is what happens. The light comes up, the objective undergoes total internal reflection. So it only excites about 100 nanometers above the cover slip. And then we can image the T cell receptor on the bottom of the T cell. And we attach an antibody which has a dye on, so we can see individual TCRs. And then we can watch them diffuse around, like I showed at the start, on the cell surface. So each of these is about 10 nanometers in diameter, but it's blurred by diffraction to a 200 nanometer spot. So we can answer questions: are they static? Do they diffuse? Are they clustered? Or do they cluster on the activation of the T cell? So the other thing, instrument that we've been building is a so-called light sheet instrument. So instead of the light coming up through the objective, we have a sheet of light that comes from the side, and we have this very fine pipette. And we hold this pipette a few hundred nanometers above the cell, and we want to shoot out protein aggregates onto the surface of the cell. And these protein aggregates interact with the receptor, in this case toll-like receptor 4, which is a receptor Nick Gay works on, and they trigger, it signals, and it forms a signaling complex And what we did is label one of the components that makes up the signaling complex. And so when we drop these aggregates on the surface of the cell in real time, we can watch these fluorescently labeled, it's MID88, forming these signaling complexes on the cell surface. So we can watch this triggering in real time. And what we're going to drop on the cell are A beta aggregates. I'm going to talk more about these later on, but these are aggregates of a protein associated with Alzheimer's disease. So first of all, I'm going to show you that we're very good at delivering molecules. We've got this very fine pipette. The computer just applies pulses of pressure, and we can shoot them out at the right patterns. So here we are. Each spot is about a micron. This is real time. We can deliver molecules onto a glass surface. And now we're going to deliver them onto a cell surface. You can see these midosomes form. You can watch them form in real time, and they disappear because they get endocytosed. So we can follow this signaling process on the cell surface in real time. If we want to get information about the structure of the cell, we can't have the molecules diffusing around, so we fix the cell. And then, as I'll explain later, we can actually locate where the molecule is with a precision of about 20 nanometers. So I just wanted to show you the type of things that we can do. So this is a T cell. It actually has these fine microvilli, these fingers on its surface, but you can't see them in a white light microscope. So what we're going to do is a super resolution imaging where we break the diffraction limit and image molecules on the cell surface with 20 nanometer resolution. So this is an experiment that we ran overnight, a bit jerky, but we're imaging the cell surface, going up and down the cell surface, and then we can reconstruct the whole cell. At very high resolution. And you can see these sort of finger-like projections coming out of this cell. So this is what a scanning electron micrograph, and hopefully you can see that this looks very similar. So what we can do now is measure the topography of the cell and ask where this T cell receptor, which is the receptor that's responsible for the adaptive immune response, where is it located on the cell surface? Are there more of these T cell receptors on these fingers, so when the cell first touches another cell, there are lots of T cell receptors, or are they randomly distributed on the cell surface? So in this experiment, we first of all, with red, label the T cell receptor, and then we come back and we re-image the cell topography. In this image, hopefully, you'll see there are lots of fingers that we couldn't see with optical microscopy. I'm happy that you can see. And you can see all these finger-like projections. And so we can ask this question, are these T cell receptors randomly distributed or localized on the microvilli? And the answer is that they appear to be randomly distributed on the cell surface. So the way that this all works, it basically, this is the idea. We have a dye, we have a green laser, and I excite the dye and yellow light comes out. And so all I have to do is look for yellow light, and I can remove all the, there's no contribution from the green. And I basically do this so sensitively, I can do it at a single molecule level. So more technically, we excite a dye molecule, which is attached to our molecule of interest. It undergoes rapid relaxation and then fluoresces. And I can do this cycle again and again and again. And it turns out I can't do it forever. I can do it 1 million to 10 million times before the molecule photobleaches and no longer becomes fluorescent. But that's enough signal. I can detect 1% of these photons. So that's enough signal for a sensitive camera to actually see a single molecule. So one fluorophore gives us 1 million or 10 million photons, and that's the basis of single molecule fluorescence. So what I'm going to talk about now is our applications to Alzheimer's disease. So in Alzheimer's disease, soluble proteins, tau and beta amyloid form these aggregates. So these proteins are interesting, they're unfolded in their native state, and they form these characteristic amyloid plaques, which are extracellular, and these intracellular tau tangles. And what's been found over a lot of research, it's not these final end products that are damaging to neurons and result in memory loss and ultimately neuronal death, but it's the small soluble aggregates on the way going from the monomer to these fibrils that are toxic. So we've put a lot of effort into developing methods to determine which aggregates are toxic in humans and their mechanism of toxicity, and actually to identify the real species that causes the disease in humans. So, there's a whole range of different diseases. Alzheimer's disease, where amyloid beta and tau form these so-called amyloid fibrils, which are these end products which have these very narrow fibrils. They're less than 10 nanometers in diameter and have this characteristic cross-beta sheet. In Parkinson's disease, alpha-synuclein, the intracellular protein that aggregates. You have this familial amyloid polyneuropathy, which is transthyretin. You have Creutzfeldt-Jacobs disease, which is prime protein, which I'll talk about briefly in this talk. In type 2 diabetes, it's amylin. So there's a very common process resulting in these amyloid fibrils that underpins a lot of these diseases. So what happens at the molecular level is you start out with monomer. The monomers form dimers and trimers, and then we've shown through doing single molecule experiments that I don't have time to discuss, that they can undergo a slow structural conversion going from these globular aggregates to these fibrillar aggregates, and these fibrillar aggregates then can grow into these long fibrils. And it seems that these smaller soluble aggregates, these globular and fibrillar aggregates, are possibly the bad ones that damage cells and cause Alzheimer's disease. And that's what we want to study. So there are lots of challenges in studying these oligomers. First of all, the concentration is exceedingly low. It's done in the picomolar range, and typically less than 1% of the molecules are aggregated, and most of them are monomer. And they're very heterogeneous in size, like I've shown you, and in structure. So we have to bridge a concentration gap. We tend to do experiments at micromolar concentrations in the lab, and actually physiologically it's picomolar concentrations. There's a reality gap. We tend to use pure beta amyloid that we buy from Sigma, and actually it gets post-translationally modified in the brain. It gets truncated and chemical groups get added. So we really want to study the real aggregates. And then lastly, there's this time gap. We study aggregations in 24 hours, and it takes decades to develop the disease. And like I showed here, a lot of experiments are done in mice, and mice aren't really like humans, but they have their brain is smaller, the ratio of astrocytes to neurons is different, and so they're not perfect models of what goes on in human beings. So our approach is we're developing quantitative methods to characterize unlabeled aggregates to understand the molecular basis of neurodegenerative disease. So we can measure the number, size, structure. We've got 2 assays that measure the toxic properties of these aggregates. We can measure how easy it is to degrade them, and we can measure their ability to seed aggregation. And I'm just going to give you one example of recent work that we've done in this area. So this is an experiment that Matthew Horrocks, who's in the audience, did a few years ago. So what is commonly used in the lab is a dye called thioflavin T. And this dye is in this, looks like this in its unbound state and is non-fluorescent. But when you put in beta sheet containing oligomers, it flattens and becomes highly fluorescent. So in the experiment that I didn't actually think would work, we got CSF, cerebral spinal fluid. So a patient with Parkinson's disease has a lumbar puncture, and you extract cerebral spinal fluid. And from our collaborators at UCL, Matthew, I think he actually went down and collected these samples from UCL and came back. And so what we did is dilute this CSF tenfold, I think, added 1 micromolar thioflavin T, and put it on one of our sensitive microscopes that I've just shown you. And what we found is we saw these small fluorescent spots. The aggregates, we think we're imaging protein aggregates in cerebral spinal fluid. Each spot is blurred by diffraction, so it's diffraction limited, but the aggregates are probably smaller. And so we started to think, well, what are these aggregates? Are they alpha-synuclein? Are they beta amyloid? What happens? Does a person with Parkinson's disease have more aggregates or bigger aggregates than a control person? patient who hasn't developed the disease. So we've had a big research program trying to understand what these aggregates are. So our sort of eureka moment was we can actually study these aggregates extracted from a human being. So it's quite impressive. We're now looking at a single protein aggregate that we've extracted from a person and just absorbed onto our cover slip and imaged. So what I'm going to show you is a series of experiments that we've done trying to characterize which of the aggregates present in CSF are toxic. So to start off, we use two assays. We have an assay where we put a whole bunch of small liposomes. So these are basically mini cells. They have a lipid bilayer arrayed on the surface, and inside each liposome there's a dye that binds calcium. And what can happen is the aggregates can temporarily permeabilize the membrane, and a calcium ion can enter. It binds the dye and becomes fluorescent. We can have thousands of these liposomes arrayed on a microscope slide and count how much calcium enters, and do this in parallel on thousands of liposomes. So we originally did these experiments on cells, but we didn't like working on cells because they responded and pumped out the calcium. So if we do this assay and we take control CSF, we see a low level of calcium entry. If we take people who are so-called mild cognitively impaired, which is on the way to developing Alzheimer's disease, so in this trial, we think 80% of these people will go on to develop Alzheimer's disease. We see a statistically significant increase in the amount of calcium. And if we take people who've got full-blown Alzheimer's disease, we see the same as the controls. So this was a set of people in Gothenburg in Sweden, all at 70, that we got from our Swedish colleagues. If we do a different assay, so in this assay we take the CSF, dilute it and put it on microglial. So these are cells that are present in the brain that are responsible for the immune response. What happens over time is they produce a pro-inflammatory cytokine called TNF-alpha, and we can measure that as a function of time. So if we do this with the control CSF, we get this basal level of increase in TNF-alpha. If we do it with the MCI, it's very similar. But if we do it now with the Alzheimer's disease CSF, we see this increased amount of secretion of TNF-alpha. So as the people are developing the disease, early on they have more aggregates that seem to be able to cause membrane permeabilization, and later on they have more aggregates that seem to be able to cause an inflammatory response. So just to show that these effects are actually due to aggregates, we can add antibodies which will bind to these aggregates and stop them interacting with the liposome. And when we have, or we can stop them interacting with the cells. So if we start with the cells first, an N-terminally binding antibody reduces the amount of TNF-alpha produced, while a C-terminally binding antibody doesn't. So if you form fibrillar aggregates, the C-terminus isn't accessible. So this suggests to us that it's the fibrillar aggregates that are present in the CSF that are causing the inflammation. On the other hand, in the calcium assay, both the N and C block reduce the amount of calcium influx, suggesting it's a more globular structure with both the N and C terminus accessible. Lastly, the receptor that's causing the inflammatory response, we used an antagonist, RSLA, which is very specific for a receptor called toll-like receptor 4, and we could show that if we add that, it reduces the amount of TNF-alpha produced. So that means that the inflammatory aggregates contain fibrillar A-beta and are signaling via toll-like receptor 4. So what do these aggregates actually look like? To do this, we do super-resolution imaging, which I'll just talk you through. So like I explained, normally everything's blurred by diffraction to about 200 nanometers. But we can image single molecules. I've shown you how we do that. And we know, and if you have a single molecule, it's also blurred by diffraction, so it looks like a 200 nanometer spot. But we actually know that the molecule must be, if it's a single molecule, it must be at the center of this spot. So we can localize where the molecule is with the precision greater than the diffraction limit. And it just depends on our signal to noise. So typically for us it's in the range 20 to 40 nanometers. So what we're going to do is replace this blurry spot with a small spot representing where we think the molecule is. And we're going to do this experiment again and again and again. And we're going to build up a super resolution image of our structure. So this is going to take us some time, but we're now going to get a super resolution image done at 20 nanometers. So the way we did this is a method that Matthew, again, developed with Eric, one of my students, and we chose an atom of DNA. So this is a sequence of DNA that's been evolved to bind aggregates. This sequence binds A-beta, beta amyloid aggregates, and it also binds synuclein aggregates. And so if we put it in solution, it's going to bind the aggregate. And what we've done is extended the sequence so it has a docking strand here, and we have an imaging strand here which has a fluorophore. So when this imaging strand binds to its complementary strand, which is bound to an aptamer, which is bound to an aggregate, which is static on the surface, we will see a spot. Then when it comes off, we won't see a spot, and then a new one binds and we see a spot. So this is the data. This is the diffraction limited image of an aggregate. This is alpha-synuclein. This is the super resolution image. This is the fibril, very blurry. This is the super resolution image. And similarly, we see the same thing with the baby. So we can image now these aggregates with a resolution of about 20 nanometers. So what we're going to do is take the CSF from these people unfortunately developing Alzheimer's disease and image all the aggregates and measure their size distribution. So this is what we see. So first of all, if we count the number of aggregates, we're slightly surprised there's no difference between the control, the MCI, and the Alzheimer's disease patients. So you don't get more aggregates as you develop the disease. But if we plot this is the length distribution. So this is the plot of the length, how the length of the aggregates varies. So if this comes up very fast, we've got a lot of very short aggregates, and if it comes up very slowly, we have a high proportion of long aggregates. So this is the cumulative frequency. So at this point, half the aggregates are less than about 50 nanometers in length. So the first thing you notice that we go out to 200 nanometers. So we've got some very large aggregates present. The gap between neurons is only about 50 nanometers. So these things are going to get stuck. And hopefully you can just see by eye that the AD-CSF comes up more slowly. So that means we've got a higher proportion of longer aggregates present in the Alzheimer's disease patients than the MCI or control. Actually, there is a difference between the control and MCI. You can't actually see it in this plot. So really to confirm this, we did atomic force microscopy. So this is a technique where we took the CSF, dried it onto a very flat surface, mica, and then you have a very fine stylus that effectively scans over the surface and can image the aggregates. And this has a very high resolution in Z. It can measure height to 0.1 nanometers. And this is what you see. So if you start up here, this is the control sample, the MCI sample, and the Alzheimer's disease sample. And as it gets brighter, it means that it comes up higher from the surface. So hopefully you can immediately see that there's clear differences by eye between the different samples. If you come down here, so that's a 50 nanometer scale bar, you see all these little aggregates in the control sample. You see these longer, taller aggregates in the MCI sample, and you see these even longer and taller aggregates in the Alzheimer's disease sample. So these people really have different shape aggregates as they develop the disease. So this is a quantification of the data. This is the height of the MCI aggregates. This is the height of the Alzheimer's disease aggregates. There are two populations, one that come half a nanometer above the mica, and some that come 2.5 nanometers above the mica. The control are very short, around 50, 70 nanometers. MCI are longer, and the Alzheimer's disease even longer. So what this all means is that as you develop the disease, the monomer becomes the oligomers, that becomes these long protofibrils that ultimately become the amyloid plaques in your brain. And these oligomers are good at permeabilizing the membrane, and this causes calcium dysregulation, which is one of the things that happens during Alzheimer's disease. And these protofibril aggregates are very good agonists. They can cause inflammation by toll-like receptor 4. So as the disease progresses, you get different high proportion of longer, larger aggregates, and you get the disease mechanism changing to becoming much more inflammatory via toll-like receptor 4. And we have a project trying to find a block of toll-like receptor 4 as a potential therapy for Alzheimer's disease. So the other really interesting thing about these diseases, so this is a review by Michel Goddard at the LMB, is that A beta propagates through the brain, appears to spread in a very characteristic pattern through the brain. Tau, which is the other protein, spreads through the brain but has a different pattern, and alpha-synuclein spreads through the brain but has a different pattern. So this gave rise to the idea that these aggregates actually spread by interconnected neurons through the brain. And so what's supposed to happen at a molecular level is this. Is this. So what happens is you have a monomeric protein and an aggregate is formed and enters a cell. This aggregate can grow by addition of the monomer, and this larger aggregate can fragment into two aggregates, and this aggregate can then leave the cell and enter another cell, and then hence you can get these aggregates spreading through the brain in this type of fashion. And this is supposed to be exponential, as I'll explain in a minute. So the problem with this idea is it sounds very nice, but there's no direct evidence of this process actually happening. So what we did is take tau. As I mentioned, tau is the protein that forms these neurofibril tangles in the brain. We aggregated in the test tube and added a dye that binds to these aggregates, then imaged it using our microscope. And what you see is for wild-type tau after 96 hours, you see these little fibrils, and you can see that they grow after 408 hours, and then you can see that they actually get shorter again as we let the experiment go. And what's happening is that they're now starting to fragment into smaller aggregates. This is a mutant of tau which is associated with frontotemporal dementia, so it is more aggregation prone, and so you can see exactly the same thing, but it's much faster, very small aggregates that grow and then fragment. So we can measure this data. This is the parrot length versus time. This is for the wild type, and this is for the mutant. And this is a fit of the data to a simple model that I'll explain in a minute. So by fitting the data, what we can do is get at the rate constant. So we're particularly interested in the rate constant for addition of monomer for the aggregate to elongate, and the rate that hasn't been measured before for the aggregate to fragment. So what we find is this is the rate constant for the elongation of the wild-type protein, and this is for the mutant. Hopefully you can see that these differ by about a factor of 100. The mutant can elongate 100 times faster, and we have this fragmentation rate now for the first time for tau, and these fragmentation rates for the wild type and the mutant are similar. We get a bunch of other rate constants that are of no interest for the moment. So what happens now is we have a fibril, it's going to elongate and break, and the two daughter fibrils are going to enter cells, and we're going to do this again and again and again, and we can work out what would happen if this was a rate-limiting process. So in a mouse, there's 70 million neurons, which only means that you have to have 22 rounds of replication of these aggregates for there to be an aggregate in every neuron in the mouse brain, and for a human it's 37 rounds. And we can actually work out what the rate of this process is if this replication is rate limiting, because it just depends on the product of the elongation rate and the fragmentation rate and the intracellular concentration of the protein. So that would be a very coarse estimate, because we think these rate constants are going to change inside an animal compared to what we measure in the test tube. But our experiments at least showed, in principle, the rate constants are fast enough to explain Alzheimer's disease. So what we can produce this plot. So like I said, it's a product of the fragmentation rate and the elongation rate. So the further I move away from the origin, the faster the aggregates replicate. So we've measured this for prion protein, and then we've measured it for the mutant of tau and wild-type tau and alpha-synuclein. And so what we can say now quantitatively is how poorer. So the idea is that these aggregates behave like a prion, but we can quantify how much poorer they are than a prion. So alpha-synuclein, for example, replicates 1,000-fold times slower than prion protein. So what I'm showing here, the only one that we could actually capture replicating is prion protein. So we can put prion protein on a cover slide, add monomer, and hopefully you can see it grows and then it fragments into two aggregates. So we can actually capture that occurring and show that happens for the first time. So what we're trying to do now is image these processes inside a cell. So this is imaging tau inside a cell. Tau binds to microtubules, so it should you see the microtubules all over the cell. This is the diffraction limited image, and this is our super resolution image, which was taken in the way I've explained to you. So what we could do is now add tau aggregates to these cells and ask what happens. So if we do that type of experiment, we see formation of lots of little aggregates fairly rapidly inside the cell. This is an antibody that just binds to tau aggregates, not monomer. And this is just to show you that we can see these very small aggregates forming inside the cell. Other data. And so we could do a time course. So this is what we see before we add the seeds. And after 7.5 hours, we've already got lots of little seeds inside the cell. And after 24 hours, the cell is jam-packed full of lots of little tau aggregates. So we now want to quantify this, see how the rate constants are different from what we measured in the test tube. And then we want to expand this towards studying real neurons to see if the same process takes place and how fast it occurs. So just to summarize this part of the talk, I've shown you that we can image aggregates from humans. We're trying to find out how they initially damage neuronal cells. We feel that this has potential for early diagnosis of the disease and the potential to monitor how effective therapies are, and we hope it will provide a basis for the rational design of new therapies. So what I wanted to just finish with is the best example of how physical methods can empower biology, which is work, like I said, which I did very early on when I arrived at Cambridge, which is on DNA sequencing. So the idea behind DNA sequencing of individuals is to understand the difference between humans and find the causes and cures for common diseases and be able to predict from your sequence your probability of developing a disease and monitor and maybe treat that before you actually develop the disease. And so it's a big challenge. You've got 3 billion bases in every cell, and every individual differs by 1 base in 1,000. And the challenge, so this was in the mid-90s, what was to develop a method that was cheap, so you could sequence in 1 day for $1,000 a whole human genome. So this story starts in the chemistry department, and then with my colleague Shankar Balasubramanian, we founded a company, Selexa, which was then eventually taken up by Illumina. So Selexa was the idea behind this was a play on a single molecule. And then Shankar thought any company that was going to do well, like Xerox, should have an X in it, so we came up with Selexa. So this is our symbol. So this is sort of a hairpin of DNA with a fluorophore. So this is how, at the time, the sequencing was done. This was a method developed by FMedSci in the biochemistry department in Cambridge. So you have DNA, and you have these bases. And what in FMedSci's method, you copy the DNA, so you have a DNA polymerase, you have all 4 bases, and you have terminators, which are either fluorophore labeled or radioactively labeled. So if you incorporate one of these terminators, you stop copying the DNA. So you're going to copy large numbers of DNA, and it's occasionally going to terminate. So it could terminate producing a very short fragment, or a longer fragment, or a longer fragment. So you're going to get a whole set of nested fragments, and then you're going to run them down a gel. The small ones are going to come out first, the long ones are going to come out later, and so you can work out the sequence of the DNA in this method. And so that was the method that was used to do the consensus human genome that was done at the year 2000 or so. And the challenge is to make this method fast and cheaper. So as I've explained to you, a big revolution in the physical method was, instead of studying 10 to the 15 molecules in a cuvette, to go down to either a single molecule with multiple fluorophores and ultimately a single molecule with 1 fluorophore. And that really it enables us to do molecule by molecule analysis. So I've shown you that we can detect these very rare aggregates and measure their structure individually. It's really the ultimate in miniaturization. And so in 1995, me and Shankar came up with a grant proposal to the BDSRC to do an experiment that we never actually got to work, which was to have a template primer of DNA, and we were going to have a polymerase, which is the enzyme that adds the nucleotides, and it was going to be fluorescently labeled, and we'll have a fluorophore attached to this template primer. And when we excited a fluorophore on the polymerase, if it was close, it could transfer its excitation to the fluorophore on the DNA, and that depended on their distance apart. So we could watch the polymerase in real time spiral up the DNA, and our signal was going to get smaller and smaller and smaller. Because DNA is helical, it would actually be a sinusoidal decay. And we couldn't really figure out how to do that experiment, but we did come up with a way of sequencing DNA much more rapidly, which I'll explain to you in the next few slides. So Shankar likes showing this slide. So frustrated by not getting this experiment to work, we did go to the Panton after work, and we did have the odd drink. So that's Shankar, well, that's me back in 1997. So I haven't changed very much. And this is the idea. So the idea is we have our template primer, and we're going to have a polymerase, and we're going to have color-coded bases, each one's going to have a different color, and we're going to organize things that we incorporate a base, and it's blocked so the next base isn't incorporated. So we have plenty of time to read which base has been incorporated in our DNA. We're going to remove the fluorescence, we're going to undo the termination, and do another cycle of sequencing. So this is the nucleotide that Shankar designed, so it has a reversible terminator and a cleavage side for the fluorophore. And so the idea is we take genomic DNA, we fragment it and attach it, so we have single molecules on the surface, that they just need to be around a micron apart, so we can resolve them, and then we're going to, in parallel, sequence all these different single molecules of DNA at the same time. So if you do a simple calculation that there are 3 billion bases in the human genome, we'll assume there are 300 by 300 single molecules in our array, and we'll assume it takes 10 seconds to image them. So you can sequence at about 10 to the 4 bases per second. So one machine would take 5 days in order to sequence a human genome. So that was the idea. It meant that we could sequence billions of bases of DNA much more rapidly than the current technology. So one of the early proof statements was actually to show that we could see single molecules of DNA. So this is DNA attached to the surface. And as I explained to you, if we have a single molecule, it should produce fluorescence, and then it should undergo this irreversible photobleaching. So most of these spots are going to fluoresce, and then they're going to turn out. Occasionally we get, by chance, 2 molecules of DNA that are closer than the diffraction limit, and so then we see 2 steps in the photobleaching, and very rarely we see 3 molecules in the same spot. So this is a bit grainy, this data, but hopefully you can see these molecules slowly disappear. You keep your eye on this. That one's gone. This one is going to go. And so this was actually for the VCs a very important statement that could actually detect single molecules, and this approach might be viable. So there's a problem with this. So typically we have these nucleotides that are labeled with dyes, and the polymerase has to operate at a concentration of about a micromolar. And only one of these nucleotides is going to be incorporated into the DNA. So if any of these other molecules stick to our surface, we can misinterpret that as incorporation. And so despite making very non-stick surfaces, that non-specific binding of the nucleotides meant it was very hard to do many cycles of sequencing. So what the company did was basically amplify the single molecules of DNA on the surface. So the thing we worried about is if we amplify the DNA, we might amplify different sequences of DNA more or less, but that turned out not to be a problem. So what you do is take the sequence of DNA you're interested in and sandwich it between two adapters. You then put it on a surface, hybridize it on a surface, and then you can get this cluster growth in this fashion on the surface. So you can take one molecule and amplify it into a little cluster of about 100 molecules of DNA. And then we're going to sequence each of the clusters. So we're going to come in with our color-coded bases. We're going to measure the color. We're going to remove the fluorescence, remove the termination, repeat the process, do this again and again and again. And the idea is that we only have to sequence 25 bases in order to map uniquely back onto the human genome. So what this looks like is all these different color spots. The current version of the instrument, that they actually have them ordered, but in the early versions, they're just randomly distributed on the surface. So if you have something like this, well on top of each other, you would simply not bother trying to determine the sequence from that. And so, like I said, after 25 cycles of sequencing, you can map this uniquely back onto the human genome. And the company actually reads the DNA from two ends of from paired ends, and that enables one to get much longer reads. So typically, the read length in the current instruments is out at 400 bases. So this is what the instrument looked like, the early instrument that Selecta first put out in 2006, which can do a billion bases per run. In 2015, it can do 1 trillion bases per run. And so we've gone from 1997, which is the Sanger-based method, where we have 10 to the 5 bases per instrument, to 2015, where we can do 10 to the 12 bases per instrument. So that's an improvement of a factor of 10 to the 7. We've gone from 100 sequencing lanes to 10 to the 9 sequencing lanes, again an improvement of the factor of 10 to the 7. And so we've gone from the cost of a human genome, which was about $1 billion, so $1 per base, up down to about $1,000, so about 10 to the 6 for cost reduction. So what can you do with this? So a whole bunch of species have been sequenced. This is the sequence of a panda. So in 2001, this consensus human genome, so this is the average sequence of everyone on the planet. In 2008, we published a paper where we sequenced an individual, an Afro-American, so that was one of the first times an individual's had their genome sequenced. And this, like I said, has opened up this idea of personalised genetics and medicine. Just to show this, in the UK there's been a big effort. So the idea is to sequence 100,000 people and follow them through their life, and you have the entire genome, and you can see what genetic, what the differences in DNA, how they predispose people to get certain diseases. And even now in the NHS, for babies with who have diseases that haven't been diagnosed, they're now going to be sequenced to find these rare mutations that might be causing disease. So it's been very widely taken up. And so I always worry about putting this slide up. So for the entrepreneurs in the audience, this is how the company was formed and the amount of money that was needed to produce the sequencing instrument. So we started off in 1995 with ideas and some very simple proof of concept in Cambridge. And then we went to a venture capital firm called Abingworth, and we got 0.6 million. So just to calibrate people, that's the type of grant that we can get from one of the research councils. So that would be a standard grant. So that's not a large amount of money to do advanced proof of concept. So this is just to show that we could see single molecules and the idea would work. Year 2000, we got 1.5 million, again from the same VC. So this is again a grant one could get from a research council. And in 2001, we opened a facility, Little Chesterford, with 12 million from Abingworth, Oxford, Bioscience, and Schroeder Ventures, and Amadeus. So 12 million is now starting to be amounts of money that would be very hard to get from research councils. In year 2004, we got another 8 million, and then in year 2005, we got another 50 million. So hopefully you can see that the amount of money that's going in is going up fairly exponentially. at this stage, we could sequence some small genomes. In the year 2006, the first commercial instrument was launched. And in the year 2007, the company was acquired by Illumina. So if you'd invested, the investors had put £170 million in to fund the work, and they received 600 million. And then everything proceeded faster with Illumina, so I haven't extended this, but now there are about 3,000 instruments in the world. So on this Illumina platform, 1 petabyte of DNA sequences was taken last year, so that's 10 to the 15 bases, and I think 90% of the world's sequences have been taken using the Illumina platform. So the reason for showing this is just to show you how fast you can go, literally go from a calculation when we've done no experiments to something that's been widely used in labs all over the world in the space of 10 years or less. And just to challenge the younger people that if they have ideas that they think might be commercial and might be able to make a difference, certainly to try and explore this. So we didn't know if this was going to work or not work. We were just very keen to see how far we could get and what might be possible. And certainly we were very surprised how well it actually worked. So you just don't know, but I think you owe it to yourself and you owe it to society to at least try and see what might be possible. So I think that's it. I just wanted to thank my group. So this is my group on the group retreat last year. This is in Chamonix. And these are the key people whose work I presented. Matthew Horrocks is in the audience, Jason and Eric, Fran, Craig and Daniel, Simone who took the fantastic AFM images, Bing and Alex who took the 3D, and a whole bunch of collaborators over the years, all based in Cambridge. And thank you very much for your attention.